All right, let's be real. Molecular inheritance can feel like the toughest chapter in biology, right?
Well, it's because most of us just try to memorize the words instead of the actual process.
So let's flip that. This chapter isn't a list of facts. It's really just one logical story.
And the secret key to unlocking that whole story? It's something called the central dogma.
Seriously, everything we're going to cover fits right into this one simple flow of information.
So, our story starts with the master blueprint for all of life, DNA.
The code's simple. It's got 4 letters. A always pairs with T, and G always pairs with C.
This rule means if you know the percentage of one base, you can figure out all the others.
Okay, so how does a cell copy this huge blueprint before it divides? It uses this process,
Called semi-conservative replication. Basically, it just keeps one old strand as a template for the new one.
And a whole crew of specialized enzymes gets the job done. Helicase unzips, polymerase builds, and ligase glues.
Now, because of how DNA is shaped, one strand gets built smoothly, but the other is built in pieces.
Okay, copy's made. Now the cell can finally read the instructions to actually build things. For that, you need 3 types of RNA working together: the message, the taxi, and the factory floor.
The message is read in 3-letter words, and it has very clear signals for start and stop.
And just how big is this blueprint? The Human Genome Project found it's about 3 billion letters long.
But here's the wild part. Only about 2% of that DNA actually codes for proteins. For years, scientists called the rest junk DNA. Turns out, it's actually running the show.
Which leaves us with this incredible question. What other secrets are still hiding in that other 98%?